Good morning, everyone. We are presenting Biotrace, an AI-powered eDNA biodiversity monitoring platform. Our solution automates species identification and biodiversity assessments from environmental DNA samples, helping researchers and conservation agencies monitor ecosystems faster, more accurately, and at scale. This slide contains template instructions and it's not part of the project presentation. Our team consists of Debabrata Majumder, Arghadeep Bose, Chayana Nandi, and Ahana Mondal, with all in expertise in software development, machine learning, bioinformatics, and data analysis. Together, we designed Biotrace to bridge the ecology and artificial intelligence gap.
Hello, I am Ahana Mondal. I am taking over the narration from here. I will be presenting the slide number 4 and 5. So my first slide presents the problem statement understanding. What is the problem that is stating here is why does this problem matter? Biodiversity monitoring is essential for conservation and environmental management. Traditional species identification methods rely on expert manually examining organisms, which is time consuming, expensive, and difficult to scale it. The environmental DNA or eDNA provides a non invasive way to detect species from water, soil, or air samples like that. However, analyzing an eDNA data requires complex bioinformatics, workflows, and expert knowledge. Without that, it is not possible. Our project addresses this challenge by automatic taxonomy identification and biodiversity assessment. My next slide is about solution proposed Biotrace eDNA platform. So we have to understand first what is Biotrace. Biotrace is an end-to-end computational platform that converts raw sequencing data into meaningful diversity insights. Users upload FASTQ sequencing files and the platform automatically performs quality control, species identification, and machine learning assisted classification and biodiversity analysis. The final output is a comprehensive ecological export generated within hours after rather than weeks. These are the sources that are used for uploading the FASTQ sequencing files by the Biotrace eDNA platform. That's it. This was my portion. My fellow teammate will continue from the next part. Thank you.
Greetings, I'm Arghadeep Bose and I'm gonna take over from my teammate in this slide and let's quickly walk through what Biotrace actually does. It is an end-to-end automated pipeline. You just upload your raw data sequencing files and Biotrace handles the rest. It cleans up the noisy raw data. It uses DADA2 to correct errors and pinpoint exact genetic sequences known as the ASVs. It matches those sequences against the global databases but also uses machine learning to identify species that aren't in the reference books. Finally, it generates a complete interactive report complete with biodiversity indices like the Chayana Index and the visual plots with just one click. No coding required, no manual steps, just upload, process, and understand. So how are we different from the existing tools? Simply put, existing tools like QIIME2 are powerful, but they are fragmented. They require manual setup, bioinformatics expertise, and they leave you to figure out the unknown species on your own. They are built for programmers, not biologists. Biotrace stands out in three clear ways. We are the first to combine DADA2 with a gradient boosted ML classifier. This cuts the number of the unidentified species by nearly 40%. While manual workflows take weeks, Biotrace delivers a complete taxonomy and biodiversity report in a single run, making it at around 10 times faster. We have created an intuitive web dashboard. We have taken the complexity out of the bioinformatics so that the field ecologists, conservationists, and policymakers can focus on protecting nature, not learning to code. In short, we are making advanced biodiversity monitoring accessible, faster, and smarter.
Hello everyone, myself Debabrata Majumder and I am the team lead for our project. I'll try to explain the solution architecture and methodology part. Now let me quickly walk you through how Biotrans actually works behind the scenes. It all starts with a eDNA sample, and this could be water, soil, or even air. This sample is run through sequencing machines like Oxford Nanopore, which give us something called FASTQ files, basically raw DNA data along with quality sources. The first thing we do is clean the raw data. We run quality checks using a tool called FASTQ and then filter out the noisy low quality reads. Next comes the real magic. We use a tool called DADA2. It removes sequencing errors and it groups similar sequences together into what we call ASVs. Now think of these as unique DNA fingerprints of each organism's presence in the sample. Now we need to know whose fingerprint is this. So we compare each ASVs against trusted global database like Silva, Bold, NCBI using a tool called BLAST Plus to identify the species. But here is a catch. Some organisms, especially rare or understudied ones, simply aren't in this database yet. That's where our machine learning model steps in. We use XGBoost, which looks at the DNS k-mark patterns and makes a smart prediction about what the organisms could be, even without a perfect database match. To see this in action, we built a ML fallback engine. When the system finds a mystery sequence that isn't in our database, it automatically routes it in the XGBoost classifier. Let me show you and run the code. Okay. As you can see here, it extracts patterns to identify almost 90% with confidence, ensuring no critical data is lost. This proves our ML model can accurately bridge the gap for unclassified species by integrating the intelligence. Biotrans turns raw complex data into actionable biodiversity insights that help researchers make research faster, data-driven decisions. Finally, everything's come together in one simple dashboard. The user just uploads their FASTag files, picks the marker gene like 16S or COI, and Biotrans takes care of the rest, delivering a clean, ready-to-use diversity report. Now let's talk about technology powering the Biotrans. On the front end, what the user actually sees and interacts with is the React.js along with Tailwind CSS for a clean modern interface and Plotly.js to build interactive charts and different graphs. On backend, Fast API and Python handles all the core processing logic. SnakeMake manages our bioinformatics workflow step by step, while Redis take care of the background task. For AI and machine learning, we rely on Python and XGBoost for our predictions, along with DADA2 for the core sequencing and processing. For storage, Postgres is our main database, along with a reference database like Silva, Bold, and CBI, which power our taxonomical identification. And finally, for deployment, everything runs on the Docker containers hosted on cloud platforms like AWS or GCP with GitHub Actions and CI/CD pipelines, keeping updates smooth and automatic. Together, this stack keeps Biotrans fast, reliable, and easy to scale, whether it's a small research lab or a nationwide biodiversity monitoring system.
Hello everyone, I am Chayana Nandi and I will be presenting today the 2 slides of our project. Building on what my friends has already explained about eDNA and biodiversity assessment, I will now discuss its real world impact and future potential. So in this first slide, I will be discussing about who benefits and how. We can see that the eDNA technology has several practical applications. It helps to ensure food and water safety by detecting harmful organisms and supports the healthy crop production and can be used to monitor diseases in humans, livestock, and wildlife. This makes eDNA valuable for governments, researchers, farmers, industries, and local communities. It is particularly useful in remote and biodiversity rich areas where traditional field surveys are time consuming and difficult to conduct. Now, moving to the next slide, my next slide, I will be presenting about business and future plans. This technology can be provided through a subscription based platform for universities, research institutes, and environmental organizations. This platform can be used to AI and machine learning tools to identify the species more quickly and accurately from eDNA data. Looking ahead, future developments include real-time biodiversity monitoring, collaboration with global university databases, and AI-powered early warning systems that can detect the biodiversity loss before it becomes severe. In conclusion, eDNA is more than just a scientific innovation. It is a powerful tool for understanding, protecting, and preserving life on the Earth. As this technology continues to evolve, it will play an increasingly important role in safeguarding biodiversity for future generations. Thank you.
In conclusion, Biotrace combines bioinformatics and artificial intelligence to transform biodiversity monitoring. By automating complex workflows and delivering actionable ecological insights, it enables faster, scalable, and data driven conservation efforts. Thank you for your attention. We'd be happy to answer any questions.